Okay, today we're going to start talking about chromatin and transcription, starting with stuff you kind of remember from Cell and Molecular and moving off into slightly more uncharted territory. Okay, meanwhile, we're going to talk about eukaryotic gene structure. Here's kind of a stripped down version of a eukaryotic promoter. Notice that it's not to scale. It's got a humongous promoter proximal region, which is really only a few hundred base pairs, and the gene itself, which is in red, is very small compared to the real gene. Notice that the red arrow moving to the right, that represents the transcript start site where polymerase will actually start copying the template. And in blue, we have the Tata box, which is within, in this case, 30 base pairs or so upstream or before or 5' to the transcript start site. So, of course, the Tata box, if it's there, is where polymerase will eventually assemble with the help of a host of transcription factors, etc. The promoter proximal region will bind proteins that can be either activators or repressors, which either facilitate or prevent from binding RNA polymerase. The enhancers, of course, can be much further away from the actual protein coding or transcribe region of the gene, as we'll see in a little while. So here's one representation of RNA polymerase trying to get itself to the Tata box. You can see that it itself does not bind directly, but actually occurs with a whole host of transcription factors. So the TF2s refers to transcription factor for polymerase 2, and they're given letters. And each one of those is actually a protein complex in and of itself. We also have the mediator, and we're also representing activator proteins that are bound to regions within the proximal promoter region that are going to facilitate assembly of the pre-initiation complex at the Tata box. Taking into account our enhancer, this is from the Cell and Molecular book, we are showing RNA polymerase, which is bound to the promoter, is about ready to get started, along with mediator, other transcription factors, but importantly, activator proteins that are bound to the enhancer, which is quite far away from the transcribe region of the gene. So we're representing this as a DNA loop. In fact, it is true that enhancers can form loops, the DNA forms loops to bring what would be really quite far away in the DNA to the vicinity of the promoter and bringing proteins and sometimes RNA polymerase along with it. We'll have more to say about these at some later date. So general terminology here. Cis-acting sequences refers to the sequences that are on the same molecule. For example, the Tata box is a cis-acting sequence because it controls transcription that will occur on the same DNA molecule as itself, but not somewhere else in the genome. Proximal promoter elements are also cis-acting, enhancers, etc. The proteins that bind there are considered to be trans factors because they come from somewhere else and can act on different molecules from themselves. So that's just one way of just thinking about DNA sequences and the molecules to which they bind. Okay, importantly, we realize we've got to get a lot of proteins there, but we've got to deal with the nucleosomes in the process. So the accessibility of these sequences depends greatly on the configuration of the actual chromatin and the organization of nearby nucleosomes. So we're going to need to be able to get all these proteins there, but we sometimes have the problem that the nucleosome is in the way. So it's very important to get the nucleosome out of the way to get transcription to occur. So we can't bind if there's proteins in the way. Most of the time, promoters are hidden from RNA pol 2 by nucleosomes. Here's a picture of chromatin with the nucleosome core particle shown. The nucleosome core particle consists of an octamer, or 8 proteins, 2 molecules each of histones H2A, H2B, H3, and H4. And the DNA is wrapped around the nucleosome core octamer, approximately taking up about 146 to 147 nucleotides of double-stranded DNA. But notice that there's part of the DNA which is not actually covered by the nucleosomes. Those are the linker regions of the DNA, and those are going to be important in terms of getting access to the DNA sequences that the proteins are going to bind. So I think I said this, H2A, H2B, H3, and H4. On average, it's about 200 base pair per nucleosome, but this is not always constant. We've got the DNA that's wrapped around the core, and the part that's not wrapped around the core is the linker. There's also histone H1, which may also be bound to the outside of the nucleosome. So the term chromatin refers to these complexes of proteins along with the DNA. Okay, shown here is a detailed view of a nucleosome viewed from the top. You can see that the DNA is wrapped around the protein core. But importantly, the proteins have extensions, the N-terminal tails, which protrude out from the nucleosome and are available for action by various modifying enzymes such as acetyltransferases and methylases, etc., as we will see. So they are exposed to the environment. Okay, so in terms of allowing access to the chromatin, there's differing states of chromatin which will depend on the types of modifications that they've undergone. For example, we'll have modifications of those histone tails that are sticking out, acetylation and methylation mainly but not exclusively. There's also DNA methylation. We also have to consider actions of chromatin remodeling complexes. And access, of course, is also dictated by the degree of compaction of the nucleosomes into higher order chromatin structures. We'll do the tail modifications first. So here's a picture that kind of represents that, where you have part of the chromatin which is nicely strung out, linkers available, nucleosomes available, a nucleosome-free region, but then a region where the nucleosomes have formed some higher order, more complicated structure in which the actual DNA is much less available than is the open structure. Yet another representation of the same idea, that genes that are actively being transcribed, the DNA is much more available than it would be in a compacted state. And there are proteins and complexes which dictate which form the chromatin takes. We're going to talk about acetylation and deacetylation in a second, but the acetylated forms of the histones are associated strongly with activated transcription, but that is a reversible process. There's also forms of chromatin where the acetylation hasn't really formed yet, but it's nonetheless accessible to get that process started. But there are also various forms of repressed chromatin which do assume higher order compaction, and that's associated with largely methylation, but also cytosine methylation of the DNA. Making a long story short, the transcription possible form has accessible DNA, relatively speaking, because it's got acetylated histones, its cytosine DNAs are not methylated, RNA polymerase can bind. The switched off version has condensed chromatin, deacetylated histones, and methylated cytosines, among other changes. So we're going to depend on various histone modifications and the proteins that create and read these. Okay, so we're going to be looking at the N-terminal histone tails, those that extend outward from the core. Okay, so we can modify these in ways that lead to opening of the chromatin or repression of it. Acetylation is associated with accessibility. So acetylation allows access of proteins to the DNA. HATs, or acetyltransferases, do this, histone acetyltransferases. Okay, this is a reversible reaction and a very dynamic one in cells, where the reverse process is catalyzed by H-stacks, or histone deacetylases. Okay. Methylation is not a plus or minus thing. Some methylations are involved in activation, others for inactivation. So whereas acetylation is almost universally associated with activation, methylation can go either way. There are other modifications that are also possible that we won't talk about specifically. One is phosphorylation, of course, and the other is ubiquitination. Okay, so H-stacks and HATs add or remove acetyl groups from the lysines and arginines. And I'm going to show you what that process looks like in terms of the actual amino acids. For example, here's histone, human histone H2B, just as an example, where I've colored in red all the lysines and all the arginines. These are the basic amino acids, which are potentially subject to modification, either by methylation or by acetylation. Okay, so it's a very basic protein, as you can see, because it's got a lot of these lysines and arginines, mostly lysines. Here's lysine and arginine, and we're talking about the amino group that's part of the side chain. These both have positively charged amino groups as part of their side chains. And they are basic amino acids because they are positively charged at cellular pH. So the action of this, you have to turn your amino acid upside down now. The amino group is now located at the top. This is lysine. Acetylation by HATs, histone acetyltransferases, involves adding a 2-carbon acetyl group to that amino group that's part of the side chain, the epsilon amino group. So that removes essentially the positive charge from that side chain and allows for a looser organization, or a less stable, shall we say, nucleosome at that position. This is, of course, a reversible reaction, which is accomplished by the H stacks. The other important modification is methylation, also of lysine, and there's 3 different types of methylation you can have, again at that amino group on the side chain. One is you can have monomethylysine, you can have dimethylysine, or trimethylysine, and these reactions are characterized, or catalyzed rather, by histone methyltransferases, abbreviated HMTs. Now, some of these have their own names. The HMT is just a generic term for the histone methylases. Okay, so it's very prevalent on basically all of them. The most important ones, most extensively modified ones rather, are the H3 and H4 tails. H2A and H2B can also be modified, but are also likely to have ubiquitin added to specific lysines. Okay, ubiquitin is a 76 amino acid protein, so this is a huge, enormous, actually, addition to histone protein if you consider the histone protein itself is less than 200 amino acids. Okay, so remember, some amino acids can be acetylated or methylated depending on the situation. We've already said this. Histone acetylation is associated with active genes, open chromatin, high levels of gene expression. Okay, some types of methylation are associated with repression and others with active transcription. For example, histone H3 lys 27 methylation and H3 lys 9 methylation are associated with repression. Okay, so here's a picture of H3 and H4 as the potential sites of methylation, acetylation, and also phosphorylation. This slide is not easy to look at because they've got red and pink, and it's hard to tell which one they're talking about. So I've got a better picture in a second. Okay, here's a histone H3 tail, and the colors are much easier to see. And you can see that some amino acids can be modified in different ways. For example, lys 9 can be either acetylated or methylated. Similarly, lys 27 can be acetylated or methylated. And these modifications, which do not occur simultaneously on all the amino acids, obviously, have different meanings. As we said a minute ago, the lys 9 methylation is associated with gene silencing, and the acetylations are associated with gene expression in general. So many different modifications and many different patterns are possible according to the gene, and the combination of what's modified changes from gene to gene and from activation state to other activation states or inactivation. They're very dynamic modifications usually. We've said this before. HATs, histone acetylase, histone deacetylase, histone methyltransferases. Okay, one thing that's important to recognize at this point is these enzymes aren't just floating around independently. They're generally part of large multi-protein complexes. And this goes for the acetylases as well as the methyltransferases. I don't think I mentioned this in class, but I will next time. There are repressor complexes, PRC1, PRC2, as an example of something that's part of a much larger protein complex, and that's associated with repression and silencing. So here's DNA methylation. This is associated with repression, where the cytosine is modified to form 5-methylcytosine. And finally, we have chromatin remodeling, which goes hand in hand with our histone modifications. And there are different pathways by which a gene or genetic region can be activated. There's no simple linear process whereby this occurs. Okay, in one important way, there are chromatin remodeling complexes, ATP-dependent chromatin remodeling complexes, which actually act directly on the nucleosomes. They can slide them along the DNA, and they can evict them or disassemble them. The modifying enzymes can actually occur as a way of recruiting the chromatin remodeling complexes. Alternatively, you can have the action of the chromatin remodeling complex followed by recruitment of acetylases, for example. So there's no simple linear pathway that describes all this. So these are, in some sense, independent processes, and yet they are mutually dependent in many cases. But they are fundamentally different because the remodeling involves moving the nucleosomes around in some way, whereas the modification on the enzymatic modification by acetylation or whatever that's basically just a modification of that particular lysine on that particular nucleosome. So chromatin remodelers, there are bunches of these, and they work in different ways. The most famous one is one called a protein complex called SWI/SNF, and this one can eject or slide nucleosomes. And these generally are activators of transcription, usually acting in the promoter region. Okay, this has a human homologue, and it's called BAF in humans, or else BRG1. So it's actually identical, well, not identical, very similar complex in the yeast versus the human, but they were discovered independently, so they have different names. Okay, but there are remodeling complexes which work in reverse that are associated with repression, and these may contain HDAC or HMT activities as well. Okay, so now we need to consider promoter architecture and the formation of nucleosome-free or nucleosome-depleted regions, and the issue of nucleosome positioning. So this slide shows a picture of a yeast gene called FO5 in its repressed state. And the +1 nucleosome is shown at the beginning of the region that will be transcribed. The TATA box region is occluded by a nucleosome. The TATA is covered up by the fact that there's a nucleosome there. So in response to inducing conditions, a lot of stuff happens, which is very schematically shown here. But the bottom line is that remodeling complexes and histone acetylases will remove those nucleosomes that are adjacent to the +1 and allow the binding of the transcriptional activator, which is FO4. So this nucleosome depleted region is very characteristic of active genes. And we need protein complexes, etc., to get them that way. So what this shows is a gene, a theoretical gene, with positioned nucleosomes. So what's being represented is each nucleosome, numbered 1, 2, 3, etc., and what the peaks are is the center of each of those nucleosomes. The idea is we will have a transcription start, and it's going to make progress through the gene, but the nucleosomes occur in this particular gene in a very ordered array and with equal spacing.